There's a strange thing that happens at almost every family gathering, every office outbreak, every school lunchroom disaster. A stomach bug goes around, half the room is doubled over, running to the bathroom, missing work for 2 days straight. And then there is that one person, the one who ate the same food, hugged the same sick kid, shared the same water bottle, and simply never got sick. Not even a little nauseous. For years, people just called this person lucky. Maybe they had a strong immune system. Maybe they just didn't touch their face as much. But scientists who study viruses started noticing this pattern too often to be random. It kept showing up in outbreak after outbreak, in different countries, in different decades, in people who had nothing else in common with each other. So they decided to actually test it properly, under controlled conditions, instead of just assuming it was chansa. And what they found hidden inside human DNA didn't just explain why some people dodge stomach viruses again and again. It rewrote what doctors thought they understood about how these infections actually work in the first place. Let's start with the virus itself, because most people don't actually know what is making them sick when they say they have a stomach bug. In the vast majority of cases, it's not food poisoning from bad chicken sitting out too long, and it's usually not the flu, even though people casually call it the stomach flu. The real culprit, more often than anything else, is a tiny, brutally efficient virus called Norovirus. It causes explosive vomiting and diarrhea. It spreads through contaminated food, contaminated water, and contaminated surfaces, and it is so contagious that it can rip through a cruise ship, a school, or hospital ward within a single day. A person can become infected from fewer than 20 viral particles. To put that in perspective, a single droplet of vomit or a speck of fecal matter invisible to the human eye can carry millions of these particles. It is one of the most efficient pathogens on the planet at finding a new host, and it has earned a reputation among public health workers as one of the hardest illnesses to contain once it starts moving through a closed environment like a ship or care facility. For decades, doctors assumed that everyone was equally vulnerable to norovirus. If you were exposed, you got sick. It was simply a matter of dose and timing, and maybe a bit of how strong your immune system happened to be that week. But in the early 2000s, researchers running deliberate exposure studies noticed something that broke that assumption completely. In these studies, healthy adult volunteers agreed to be given a measured dose of norovirus under strict medical supervision so scientists could study exactly how infection begins, progresses, and clears. Everyone in the study received the exact same amount of virus. Same strain, same dose, same conditions, same monitoring. If exposure alone caused illness, every single volunteer should have gotten sick within a day or two. They didn't. A significant portion of the volunteers, sometimes as much as a third of the group, never developed the infection at all. Their bodies didn't just fight off the virus quickly and recover with mild symptoms. The virus never took hold in the first place. No symptoms, no detectable viral replication in their stool samples, nothing. It was as if the virus had knocked on the door and the door simply wasn't there to begin with. Researchers reran the experiment with different strains, different labs, and different groups of volunteers recruited from different regions. The pattern held every single time. Roughly the same fraction of people, no matter where they came from or what their general health looked like, seemed to be biologically untouchable to certain strains of norovirus. This wasn't about hygiene habits or willpower or a naturally strong immune system fighting off illness after exposure. Their immune systems were never even called into action because the virus couldn't infect their cells to begin with. It was blocked at a much earlier, much more basic stage than anyone had considered. That is when the investigation shifted away from the immune system entirely and towards something far more basic, the surface of the human gut itself. To understand what scientists eventually found, you have to understand how a virus actually gets inside a cell in the first place. A virus cannot just barge into any cell it happens to touch. It needs a specific docking point, a molecular shape, sitting on the surface of the cell that the virus can lock onto, almost like a key that has to match a very particular lock. Once it locks on, it can force its way inside and hijack the cell's own machinery to start making thousands of copies of itself. Different viruses use different locks scattered across different tissues in the body. Some viruses target receptors deep in the lungs, which is part of why respiratory viruses spread the way they do through coughing and breathing. Norovirus, it turns out, targets a very specific set of sugar molecules that sit on the surface of cells lining the small intestine. Molecules that most people have never heard of unless they happen to study biochemistry. These sugar molecules are not part of some rare or exotic biological system built just for this purpose. They belong to a well-known biological pathway involved in something called blood type antigens, the same general family of molecules responsible for the differences between blood type A, B, AB, and O that most people learn about in school. But this particular version of the pathway isn't just found in blood. It's found on the surface of cells throughout the digestive tract, in saliva, and in several other bodily secretions. Whether or not a person produces these particular sugar structures on their mucosal surfaces is controlled by a single gene, and it is this one gene that turned out to be the missing piece connecting everything researchers had observed. The gene is called FuT2. It carries the instructions for building an enzyme called fucosyltransferase 2. This enzyme's job sounds almost too simple to matter. It attaches a small sugar molecule called fucose onto certain proteins sitting on the surface of mucosal cells, and it performs the same task in saliva and other secretions throughout the body. When this enzyme is active and working the way it's supposed to, it creates a specific, recognizable sugar pattern on the surface of those cells. This raises an important and slightly humbling point that is worth sitting with for a moment. None of this happened because ancient humans understood genetics or viruses or sugar receptors. Nobody chose to be a secretor or a non-secretor. And no population consciously evolved this trait on purpose through some kind of collective decision. It happened purely through the brutal and different math of survival across thousands and thousands of years. In any given generation, in any place where a virus using this particular doorway was killing or severely weakening people, individuals who happened to carry a broken copy of FuT2 were simply more likely to survive, more likely to reproduce, and more likely to pass that broken copy on to their own children. Repeat that quiet process over hundreds of generations, largely invisible to everyone involved. And a once rare mutation can become common enough that today, roughly 1 in 5 people you pass on the street carries it, walking around with a form of built-in resistance to viruses they've probably never even heard of and will never know they're protected against. This discovery has also completely changed how scientists approach norovirus vaccine development, which has historically been one of the most frustrating and stubborn challenges in all of virology. Vaccines generally work by training the immune system to recognize and fight off a pathogen ahead of time, but that assumes the pathogen can actually get into the body's cells to trigger that recognition process during a natural infection, or that a vaccine can convincingly mimic that interaction closely enough in a lab setting. Because norovirus susceptibility depends so heavily on this one genetic doorway, and because different strains use different doorways scattered across the population, researchers now understand that a single universal norovirus vaccine may need to account for secretor status from the very beginning of the design process. A vaccine tested primarily on secretor volunteers might show promising, encouraging results in early trials while offering a completely different level of protection or an entirely different type of biological interaction in non-secretor individuals whose cells simply don't present the virus the same way in the first place. This genetic wrinkle is a major part of why, even decades after norovirus was first identified as one of the leading causes of stomach illness worldwide, there still isn't a widely available vaccine for it the way there is for so many other common childhood and adult illnesses. It's worth pausing here to be really clear about what this discovery does not mean, because genetic stories like this one tend to get flattened and oversimplified once they spread outside of scientific circles. Being a non-secretor does not make someone immune to all stomach illness in general. It does not protect against food poisoning caused by bacteria like Salmonella or E. Coli, which use completely different mechanisms to cause disease and don't rely on this sugar doorway at all. It does not protect against every strain of norovirus either, only the ones that specifically depend on the FUT2 sugar pathway for entry into gut cells. And it certainly does not mean secretors are somehow biologically inferior or unlucky compared to non-secretors. Being a secretor comes with its own set of trade-offs, and in some contexts, researchers are still exploring its own protective advantages against completely different pathogens that specifically target non-secretor biology instead. Nature rarely builds a system where one group gets all the advantages and none of the downsides, because a species that worked that way would be dangerously fragile against whatever new threat came along next. It is almost always a trade, protection against one threat in exchange for a bit of vulnerability to another, spread unevenly across a population, so that no single pathogen can ever wipe out an entire species all at once. That, in a strange and almost comforting way, is the deeper lesson hiding inside this one small, unassuming gene. Human genetic diversity isn't a flaw or a random accident that evolution somehow failed to iron out. It is a survival strategy, written across millions of individual bodies, quietly ensuring that whatever pathogen shows up next, some fraction of us will already have a lock that virus simply can't pick. Researchers are still uncovering new layers to this story, and the field is moving faster now than it ever has before, thanks to cheaper genetic sequencing. Some scientists are now investigating whether secretor status could eventually help explain differences in how people respond to other infections entirely, including certain respiratory illnesses, since some pathogens beyond norovirus are now being studied for similar sugar-based attachment mechanisms hiding in different tissues of the body. Rotavirus, another extremely common cause of severe stomach illness, especially in young children, has also been found to use related sugar receptors in some strains, meaning secretor status may partly explain why some children get hit hard by rotavirus while others in the same household barely show symptoms at all. Other researchers are exploring whether a person's FUT2 status could one day be used as a simple, practical piece of medical information, similar to how blood type is already used in hospitals today, helping predict individual risk during outbreaks in nursing homes, daycare centers, cruise ships, and other high-exposure environments where a fast-moving stomach virus can be genuinely dangerous for elderly or medically vulnerable patients. It's a small but telling example of where genetics and everyday medicine are slowly starting to merge together, moving away from broad, one-size-fits-all assumptions about disease and toward a much more individualized understanding of why two people exposed to the exact same virus in the exact same room at the exact same moment can end up with two completely different outcomes. It's worth going back to those original exposure studies for a moment, because the way they were conducted tells you a lot about how careful this kind of research has to be. Volunteers weren't just handed a cup of virus and sent home to see what happened. They were housed in monitored clinical units for days at a time, with medical staff checking on them around the clock, tracking every symptom, every temperature spike, every stool sample, and every blood draw with painstaking precision. This kind of research is expensive, slow, and heavily regulated for good reason, since deliberately infecting a healthy volunteer with a virus is not something any ethics board approves lightly. But it was exactly this level of control that made the genetic pattern impossible to ignore. In a real-world outbreak, there are 100 different variables that could explain why one person got sick and another didn't. Different amounts of food eaten, different exposure times, different immune histories from past infections. In a tightly controlled clinical trial, almost all of those variables get eliminated. Everyone gets the same dose, the same strain, the same monitoring, the same environment. When roughly the same percentage of people came back completely uninfected trial after trial, year after year, researchers knew they weren't looking at noise. They were looking at a real, measurable biological divide running straight through the human species. The scientists who first traced this resistance back to FUT2 didn't stumble onto it by accident either. It took years of painstaking comparison between infected and uninfected volunteers, cross-referencing their genetic profiles, their saliva samples, and the specific sugar patterns present or absent on their gut tissue. Early researchers noticed that resistant volunteers all shared something in common in their saliva testing, a complete absence of certain blood group antigens that almost everyone else had in abundance. That observation was the thread that eventually led back to the FUT2 gene itself. And once the enzyme's role in building those exact antigens was confirmed, the entire picture clicked into place almost overnight. It's a good example of how major discoveries in genetics rarely happen in one dramatic breakthrough moment. More often, they happen slowly through years of unglamorous lab work, cross-checking small details until a pattern that seemed like a coincidence turns out to be the whole explanation. Outbreak investigators eventually started applying this knowledge outside the lab too, in real-world settings where norovirus does the most damage. Cruise ships are a favorite case study among epidemiologists precisely because they create something close to a natural experiment. Thousands of passengers confined together, eating from the same buffets, touching the same railings, using the same shared spaces for days at a time. When a norovirus outbreak hits a ship like this, investigators can track exactly who got sick and who didn't with far more precision than they could in a sprawling city outbreak. Time after time, roughly the same proportion of passengers walked away completely unaffected, even after prolonged, repeated exposure to a highly contagious strain circulating through the ship's air and surfaces for days. Hospitals and long-term care facilities show similar patterns during their own outbreaks, which matters enormously for infection control planning, since it means a facility can never assume that improving hygiene alone will bring an outbreak to zero. A meaningful fraction of any exposed population was never going to catch that particular strain in the first place, no matter how thoroughly the surfaces were cleaned. There is also a growing interest in what this means for at-home genetic testing, now that consumer DNA kits have become so common and affordable. A handful of research groups have started flagging FUT2 status as a data point worth including in expanded reports, right alongside more familiar traits like ancestry breakdowns and caffeine metabolism. It's a strange thought to sit with, that somewhere buried in a spit sample mailed off in a small plastic tube is the answer to a question most people have wondered about their entire lives without ever having the words to ask it properly. Why does everyone else get sick, and I never do? So the next time everyone around you is sick with a stomach bug and you somehow feel completely fine, it might not be luck, and it might not be your immune system putting up some kind of heroic, invisible fight behind the scenes. It could be something written into your DNA long before you were even born. A tiny variation in a single gene that quietly decided, on a purely molecular level, that this particular virus was never going to get through the door. It's a small, strange, and oddly humbling reminder that the human body is still full of hidden defenses we are only just beginning to fully understand, and that sometimes the difference between getting violently sick for 2 days and walking away completely unaffected comes down to a few missing sugar molecules on the surface of a cell you will never see, feel, or even know exist, quietly working in the background every single day of your life without ever asking for credit.